Hello, and a very warm welcome to everyone joining us for today's Select Science webinar titled Optimizing Nuclei Quality Assessments with Automated Cell Counting. My name is Lawrence Howes, and I'll be moderating today's presentation. I'm delighted to be joined by our guest speaker, Seungwon Jo, Assistant Researcher at Logos Biosystems. This webinar will demonstrate how to streamline nuclei assessments and choose optimal fluorescent dye combinations. In addition, you'll learn how to integrate the different dyes and optimize advanced automated cell counters to ensure the reliability of your results. There'll be some poll questions popping up during the webinar, and if you could answer these when possible, that'd be greatly appreciated. Following the presentation, we'll have time for a question and answer session. So if you have any questions throughout the webinar, please feel free to submit these in the box to the right of your screen. So without further delay, I'd like to hand over to our speaker. I'd like to thank them again for presenting for us today.
Good morning, good afternoon, and good evening, everyone. Welcome to today's webinar on optimizing nuclei quality assessment with automated cell counting. I am Seungwon Jo. I usually go by Joe, who is a researcher of Logos Biosystems, part of Aligned Genetics. All right, let's move on to today's content. First, we'll focus into today's topic. Next, we'll explore the criteria used to assess nuclei quality. And then we'll introduce the LUNA-FX7 automated cell counter. Following that, we'll walk through the step-by-step procedure for conducting nuclei quality assessment. And then we'll show you analyzed results obtained from nuclei quality assessments. And we'll address common troubleshooting scenarios encountered during nuclei quality assessment. Finally, we'll summarize today's presentation before moving on to the Q&A session. Okay, our company, Aligned Genetics, was founded in 2008, so we were established 16 years ago. With a team of 75 dedicated employees, we operate in over 115 countries and collaborate with 129 distributors worldwide. And we own two brands, Logos Biosystems and GeneScale. And we are proud owners of 43 patents. Additionally, we have subsidiaries in the USA and France, with our headquarters based in South Korea. Okay then, let's talk about today's topic, nuclei isolation. So it is a crucial step in various research fields, such as molecular biology, genomics, and cell biology research. Nuclei isolation involves extracting cell nuclei from biological samples like tissues or in vitro cell samples. These isolated nuclei provide pure and concentrated genetic materials. The process overall involves lysing intact cells, followed by a washing step and filtering to obtain the extracted nuclei without contaminations from the biological samples. And nuclei quality assessment is crucial for several reasons. First, you can enhance accuracy, so it leads to more reliable outcomes. Second, you can ensure purity by preventing contaminations from external DNA sources. Third, it maintains consistency. It is crucial for comparative studies like genomic comparisons. Last, and perhaps the most important part, it promotes cost efficiency by minimizing low-quality data and avoiding expensive processes like sequencing. So ultimately, ensuring high-quality nuclei sets the foundation for successful outcomes in downstream applications. Before we get further into nuclei quality assessment, let's first introduce our LUNA-FX7 automated cell counter. So this advanced imaging tool offers precise cell enumeration and reliable cell size measurement, along with high throughput capability for rapid imaging using the variety of cell counting slide options. With both bright field and fluorescence imaging options, LUNA-FX7 ensures exceptional autofocusing performance, and it can provide clear and sharp images crucial for accurate data analysis. Its intuitive software provides a user-friendly interface and protocol flexibility, which can simplify the process of navigating and adapting to various sample types. Okay then, let's watch the video introducing the Luna FX7. First, you can easily insert your slide and focus your cells using the autofocus button. Once autofocusing is complete, you can simply press count to proceed with cell enumeration. And after counting, you will receive your results. And in the histogram and gating section, you can adjust the bars under the histogram to change the cell size range. This allows you to assign different size ranges. And finally, you have the option to save your data either on a thumb drive or the built-in SSD for future use. And as I said, we offer a variety of cell counting slides capable of holding up to 8 samples. Most slides have a sample loading volume of 10 microliters per chamber, except for the one-channel slide. One-channel slide has a volume of 50 microliters per chamber. The one-channel slide provides the highest analyst volume and field of views, FOVs, compared to others. So if you want to focus on one sample, one-channel slide is your compared to others. So if you want to focus on one sample, one-channel slide is your option. The 8-channel slide can hold up to 8 samples, while 3-channel and 2-channel slides can hold up to 3 and 2 samples, respectively. 2-channel slides are the most commonly used option and hold up to 3 and 2 samples, respectively. 2-channel slides are the most commonly used option, and additionally, we offer a reusable slide, which is both eco-friendly and budget-friendly option. Reusable slide, which is both eco-friendly and budget-friendly option. So you have the flexibility to choose the cell counting slide options that best suit your needs. However, slide bottom flatness is a common concern raised by many people when it comes to using slides for imaging. During the imaging process, image quality can be compromised due to the uneven bottom surface, causing focal plane issues. The Luna FX7 automated cell counter can effectively address slide bottom flatness issues with its superior autofocusing algorithm. This algorithm ensures high-quality imaging by accurately finding the best focal plane, even on uneven surfaces. In turn, users can conduct high-throughput imaging and simultaneous imaging of multiple samples with confidence. Coming back to nuclei isolation. So we have conducted nuclei isolation for both in vitro cell samples and tissue samples. The overall procedures for both include slices, washing, staining, and automated cell counting. For in vitro nuclei isolation, we used homemade buffer. And we used both the homemade buffer and the 10x Genomics nuclei isolation kit for tissue nuclei isolation. Effective filtering and debris removal were crucial factors for successful tissue nuclei isolation. Finally, we used Luna FX7 to analyze the quality of our samples. And when assessing nuclei quality with automated cell counters like Luna FX7, we consider 4 key criteria. First, yield. So yield is crucial for any kind of downstream applications. So basically, you need enough number of samples to work with. Second is size, as nuclei should be significantly smaller than intact cells, typically reduced by 40 to 60%. From this montage, you can see a significant decrease in size compared to intact cells. Bright field images can help evaluate remaining cytoplasm content. So from this montage, incompletely isolated nucleus has remaining cytoplasm around it, whereas complete nucleus typically has oval shape without cytoplasm. Lastly is color. So in fluorescent mode, stains like AOPI or ethidium homodimer should appear red, while trypan blue in bright field mode appears blue. So by using automated cell counters, we can assess nuclei quality based on yield, size, bright field images, and colors. Okay then, you could wonder then why nuclei express red or blue colors when stained with different dyes. So here's the list of dyes we used for nuclei quality assessment. Let's focus on acridine orange, AO, first. Unlike other dyes, AO is membrane permeable, allowing it to enter both live and dead cells. And acridine orange can bind to nucleic acids. And upon binding to nucleic acids, it can emit green fluorescence under fluorescence microscopy. On the other hand, propylium iodide, PI, and ethidium homodimer are impermeable to cell membranes, and they can specifically bind to nuclei. Both PI and ethidium homodimer can emit red fluorescence upon binding to DNA. AUPI or AO ethidium homodimer dye combinations are commonly used dyes to distinguish between live and dead cells based on their permeability and quenching effect. So in this context, a quenching effect means that the PI or ethidium homodimer signal can absorb the AO signal. And this can prevent bleed-through or double positive results. Therefore, staining nuclei or dead cells red overall if you use AOPI or AO ethidium homodimer dye combinations. And last, trypan blue. Trypan blue can bind to proteins and nuclei, and TB can stain dead cells or nuclei blue. And this can be visualized under bright field microscopy. That being said, let's discuss the trypan blue stain protocol first. So with trypan blue stain, you can simply mix your sample with TB, trypan blue, in a 1:1 ratio. Load your samples onto a cell counting slide and count them using a Luna FX7, and you can use the default protocol. Dead cells or isolated nuclei are indicated by the dark color. So for example, in this montage here, let's say you have 50% viable cells. Your results should resemble this image, with dead cells appearing dark and live cells transparent. Okay, and let's get to our UI. So if you turn on the Luna FX7, and if you choose the bright field cell counting option, you'll be able to see cell counting and viability mode. So under the cell counting viability mode, you can go to the protocol tab. And under the protocol tab, there is default protocol. So usually, default protocol is working well with nuclei samples. And let's say you have working well with nuclei samples. And let's say you have successfully isolated nuclei, stained with trypan blue, imaged using bright field modes. With trypan blue stain, you will notice differences in color and morphology. Intact cells typically show clearer and sharper edges, which makes them easier to distinguish. On the other hand, nuclei samples may have slightly dimmer edges. In the analysis results, all nuclei may be tagged as dead cells, which appears dark. However, we do not recommend using trypan blue with nuclei samples. And there are several reasons why we do not recommend using trypan blue stain for nuclei. So there are several reasons. The nuclei isolation process can introduce debris or aggregation, which may affect the accuracy of staining results overall. Trypan blue staining may non-specifically bind to various particles, which can cause inaccurate data analysis. Furthermore, nuclei typically have relatively dimmer edges, which can make them harder to distinguish edges. This can also lead to inaccurate data analysis overall. Therefore, we recommend using fluorescent dyes such as AOPI or AO ethidium homodimer, which can mitigate these issues. Speaking of fluorescent dyes, let's cover fluorescent dye staining protocol when using AOPI and AO ethidium homodimer staining. When using these dyes, you can simply mix your fluorescent dyes with your cell samples in a 1:9 ratio. Load your samples onto a desirable cell counting slide and count the sample using LunaFX7 with the default protocol. Dead cells or nuclei will be indicated by the red color. So live cells will express green color from acridine orange, while dead cells will express red color from propylium iodide or ethidium homodimer. This red color can absorb the green color from AO based on the quenching effect. So it allows you to easily distinguish between live and dead cells or nuclei samples in your sample. And then at this point, you could wonder which ones are better between PI and ethidium homodimer. Both propylium iodide and ethidium homodimer are viable options for staining nuclei. But nuclei labeled with ethidium homodimer will generally exhibit a dimmer signal compared to PI. In the provided montage, both nuclei samples stained with both dye combinations are shown. AOPI expresses a higher signal overall, whereas AO ethidium homodimer shows a dimmer signal. However, LunaFX7 can successfully tag either one. So if you only have ethidium homodimer available, it's acceptable. But if you have access to PI, we highly recommend using PI. And here's another reason why we recommend using PI over ethidium homodimer. Some researchers may want to store their nuclei after fixation. However, when using fixed nuclei, both PI and ethidium homodimer may show a diminished signal. Especially ethidium homodimer can be almost invisible when using the default protocol. To address this issue, you can consider increasing the exposure level to 8 for a sufficient signal. In summary, for effective nuclei assessment, the recommended dye combination is AOPI. Therefore, all the data presented from now on are from samples stained using AOPI. Okay, then let's move to our UI. So in our UI, you will be able to find fluorescent cell counting option. And after staining your samples with AOPI, and if you want to use fluorescent cell counting option, there's a caveat. So when selecting fluorescent cell counting, there's two different modes. One is cell lines and primary cells. Another is cell lines and primary cells advanced mode. For simplicity, we refer to them as basing mode and advanced mode. In most cases, there are negligible differences between the two modes. However, advanced mode uses an improved algorithm overall, and it enhances cell counting accuracy. The most significant differences in this case is declumping performance. While basing mode may count clumped nuclei as one large object, advanced mode successfully separates clumped nuclei. Therefore, we recommend using advanced mode for improved accuracy and declumping performance when performing nuclei quality assessment. And let's say you chose advanced mode, and you will see a default protocol under the protocol tab. In most of cases, default protocol worked well with nuclei samples overall. And when using fluorescent imaging, there are several factors should be considered when assessing the quality of nuclei. So here's the montage showing bright field, fluorescent, and tagged images after analysis. The green circle indicates live intact cells, and this should be disregarded when assessing nuclei. The red circle highlights dead cells or nuclei and is crucial to observe the overall morphology. So you need to check if the cytoplasm is completely removed from nuclei. Successfully isolated nuclei typically exhibit an elliptical shape, while damaged nuclei may appear irregular. And in this case, debris is not a concern because it will not emit a fluorescent signal, meaning any debris present in the sample will not be tagged as objects. And knowing this criteria, and let's say you have successfully isolated nuclei imaged using fluorescent mode. In the bright field images, it should be easy to distinguish between intact cells and nuclei. You can ensure there's no remaining cytoplasm or irregularly shaped nuclei. In the fluorescent images, green signals should only be visible from intact cells, while red signals should originate from isolated nuclei. Moving to tagged images, you will see a significant change in viability from almost 100% to almost 0% after nuclei isolation. Let's move on to our results. And these montages show tagged images from LUNA-FX7 after analyzing isolated nuclei samples from various cell lines. We use homemade buffer for all of these samples without debris removal buffer, and all samples were stained with the AOPI combination. In vitro nuclei extraction was successful, and the samples were analyzed using the LUNA-FX7 automated cell counter. Let's take a closer look into HL60 cells, which are suspension cells. Compared to adherent, let's take a closer look into HL60 cells, which are suspension cells. Compared to adherent cells, isolating nuclei from suspension cells, like HL60 cells, was relatively straightforward. There are some debris visible here, but it's not overwhelming. However, extracting nuclei from adherent cells, such as HeLa cells, was relatively harder for us. So we had to optimize surfactant concentrations and harder for us. So we had to optimize surfactant concentrations and incubation time to effectively isolate nuclei from adherent cells. And if you encounter the similar issue like us, here's a tip using size gating and histogram analysis. In the example provided, from left to right, you will see nuclei samples extracted using decreasing surfactant concentrations. Lower surfactant concentrations may result in more samples with remaining cytoplasm, while higher concentrations can lead to irregularly shaped nuclei, which are smaller. Can lead to irregularly shaped nuclei, which are smaller. To address this, use the size histogram. So after completing your data analysis with your sample, you will receive size histograms within your results. Based on size, you can disregard nuclei with remaining cytoplasm or irregular shaped nuclei, which should be either larger or smaller than your average sized nuclei. And then you can compare your analyzed images after adjusting your size range, and you can decide whether your image is good to go or not. Okay, moving on to tissue samples. Here are examples of isolated nuclei from various tissues, including mouse brain, lung, liver, kidney, small intestine, and heart. For brain, lung, and liver samples, 10x genomics buffer was used, while we used homemade buffer for kidney, small intestine, and heart samples. All samples were stained with the AOPI combination, and as you can see, these nuclei were successfully tagged and analyzed using the LUNA-FX7 automated cell counter. And let's take a close look into kidney. We obtained the nuclei from mouse kidney using our homemade buffer. And the isolation process was successful overall, and the isolated nuclei had excellent clarity and quality with relatively lower debris present. And here's the liver sample, and we used 10x genomics isolation kit to extract nuclei from liver sample. And isolating nuclei from tissues with the 10x Genomics kit should be straightforward if you follow the instructions they provide. However, with liver samples, we had relatively higher amount of debris in the sample. And after nuclei extraction, we noticed there is generally more debris present in the sample compared to other tissues. And one can decide whether this level of debris is acceptable or not after imaging and analysis. However, if you experience difficulties with debris removal, filtering and washing are essential steps to eliminate debris from the sample. So in this montage, you can see filtering and washing can effectively reduce debris. However, it's important to understand that excessive washing can compromise nuclei integrity, and that can lead to nuclei aggregations and a decrease in overall nucleus count. Therefore, it is essential to find the optimal balance between debris removal and nuclei preservation. In our experience, washing once was enough for washing out most of debris out. So you can proceed with more washing if it's necessary, but as I said, you can find the balance between debris removal and nuclei preservation. And if filtering and washing were insufficient to remove all debris from your samples, here are some troubleshooting tips. First, reducing tissue mass can significantly mitigate the issue. So reducing tissue mass can mitigate most of issues you may have during nuclei isolation process. So if you have any issues, the first approach I would do is reducing tissue mass. And careful filtering with a nylon mesh filter can be beneficial. And if you're using the 10x Genomics nuclei isolation kit, you can use a higher volume of debris removal buffer, and that can help in effectively removing debris from your samples. Next is aggregation. So if you're experiencing issues with aggregations, reducing tissue mass again can help mitigating aggregation. And moreover, aggregation typically occurs due to integrity issues following the overall nuclei isolation process. As nuclei isolation process involves lysis, it can damage your cells and damage even your nuclei. Therefore, shortening the incubation time and lowering surfactant concentration can minimize nuclei damage and preserve sample integrity. And then decreasing the washing step with centrifuge can also minimize damage to your samples, and in turn, it can reduce aggregations. As I mentioned before, washing step with centrifugation can damage your cells, and that can lead to aggregation in your samples. The last issue is low yield. The most important factor for low yield issue is your buffer. So using PBS alone can result in low yield overall. To prove this, we ran the simple testing to compare yield differences when storing nuclei in PBS with BSA and PBS alone. The graph on the right shows the relative nuclei concentration over time. These samples were stored in the same tubes, and the number of nuclei was counted to see the change in concentration over time. The results suggest that samples need to be handled as quickly as possible, as nuclei can bind to the surface of the tubes. So fast sample handling can prevent losing nuclei. And then storing the sample in PBS with BSA can decrease the concentration by about 20% within 30 minutes. However, storing the sample in PBS without BSA can result in 60% loss within the same time frame. Therefore, using BSA is important to protect your samples. And if BSA is unavailable, FBS can serve as an alternative, or you can simply use cell culture media. And then certain cells, such as mouse hearts, are known for their low yield, so please check your sample information. And then moreover, you can minimize the washing steps during sample preparation to keep your sample yield high. Okay, to summarize today's presentation, we highly recommend using the AOPI combination for all situations. If AOPI is unavailable, ethidium homodimer can serve as an alternative. However, when using ethidium homodimer on fixed nuclei, signal intensity issues may arise, and it can potentially lead to inaccurate data analysis. To mitigate this, you can adjust the exposure level. However, AOPI is the best dye combination. And in cases where cost is a concern, trypan blue staining may be considered. However, we cannot recommend using trypan blue due to its tendency to non-specifically bind to aggregates and debris. And this can also result in inaccurate outcomes. So overall, AOPI combinations paired with fluorescent imaging provided the best results when assessing nuclei quality. And in conclusion, nuclei isolation is a time-consuming and delicate process. And ensuring good quality control of nuclei is essential for downstream applications such as sequencing. Automated cell counters like the LunaFX7 can offer a straightforward solution for nuclei quality assessment. That wraps up today's presentation.
Thank you for that very informative presentation. It's now time for our question and answer session. So if you do have any further questions, please send those in. You can submit these by typing into the questions box to the right of your screen. So to begin with, we had a few questions come in. The first one we've got for you, Joe, is there an issue with distinguishing between dead cells and single nuclei?
Okay, there's no complete solution for this matter. What we can recommend is using size histogram. You'd get the size histogram after analysis done. There should be different peaks appearing in your histogram if you have considerable amount of dead cells in your nuclei samples. In our experience, size histogram was effective for distinguishing objects based on their sizes. So I would try using size histogram.
Excellent, thank you. So the next question we've had in is, what are the concentration ranges for the LunaFX7?
Our LunaFX7 has detectable concentration range of 1 × 10⁴ to 5 × 10⁷ cells per milliliter, and optimal concentration range of 5 × 10⁴ to 1 × 10⁷ cells per milliliter. Nuclei should fall between these ranges. However, you could experience the issue when there is significant level of debris in your sample, which can compromise overall performance. So please use good quality sample for the optimal performance.
Great, thank you for clarifying that for us. So the next question we've had in is, which modes do you recommend between basic mode and advanced mode under FL counting option when counting intact cells?
As long as you are using good quality sample, using either mode does not affect the overall performance. Within the presentation, we showed you the montage showing the performance difference between the two modes. And as I said, please use advanced mode if you have compromised samples with aggregates or a good amount of dead cells present.
Excellent, thank you very much. We've had another question in. So what should I do when the LunaFX7 fails to tag properly?
There are several reasons that can cause this failure. First reason would be signal. So low dye signal during imaging can cause compromised performance overall. So try to increase exposure level, or you can increase your incubation time with your dyes. Second can be autofocusing failure. In this case, you can try offset function in LunaFX7, or you can do AF based on different channels. So under the settings in the LunarFX7, you can choose the primary channel that you will be using for autofocusing. When you using nuclei samples, there should be predominantly red signals in your sample. So we would try using red channel as a primary channel for autofocusing. And the last point would be size. So widening the size range can also help to tag your nuclei. But I would be careful with this approach as this can result in including irregularly shaped nuclei, and these nuclei are usually smaller than normal complete nuclei.
Great, thank you, Joe. So yes, the next question we've had in is how to formulate the lysis buffer and determine detergent concentration for a homemade kit.
For this question, it depends on your sample. In case of in vitro cells, the rule of thumb is suspension cells were relatively easier to extract nuclei with lower detergent concentrations and shorter incubation times. However, samples like adherent cells or tissue samples were relatively harder to extract nuclei, and these required higher detergent concentrations and longer incubation times. You can test your results using different conditions. In our case, we used 0.025% of turgitol with suspension cells for 5 minutes and 0.075% of turgitol to adherent cells for 20 minutes.
Great, thank you very much. Just wait for a bit of time for a few more questions to come through. 2 seconds. Please feel free to keep sending those questions in, guys. Much appreciated. While we're just waiting for some questions to come in, I'll just read a little bit more about Joe. So he's an assistant researcher at Logos Biosystems in Anyang, South Korea. He's got a passion for science innovation, and he's actively conducting experiments using Logos Biosystems products. In addition to his experimental work, he plays a crucial role in generating application notes, technical notes, and white papers. And he aims to provide customers with the essential knowledge and insights for achieving success in their scientific endeavors. We've had another question come in. Excellent. So nuclei fluorescent sorting should be considered as a powerful purification method. LunarFX7 could be used to assess the sample counts and quality before and after sorting. Have you tried this?
We have not tried to use LunarFX7 to assess sample counts and quality before and after sorting. But we think that LunarFX7 can be a great option to assess the sample counts. And after sorting, I would assume there would be less debris present in the sample. And with LunarFX7 cell counter and low amount of debris, your assessment can be quite successful. So I can confidently tell you that LunarFX7 can be very effective.
Great, thank you, Joe. I'm just going to leave a bit more time for you guys to send some more questions in. So keep those coming in if you can, if you've got any more. Okay, so we're going to wrap it up there. I'd like to thank our expert speaker, Joe, for the very informative presentation and discussion. And a big thank you to everyone for joining us online. We hope you found this a worthwhile session. If you have any further questions, please feel free to email me at editor@selectscience.net, and we'll follow up with your questions for our speaker. Remember, you can download related resources in the tab to the left of your screen, and this includes the certificate of attendance. If you'd like to listen again to today's webinar or invite a friend to listen, it'll be available to watch on demand in just a few days. Goodbye, and thank you once again for joining us.